Welcome students. So if we keep red blood cells and a plant cell in three different conditions, then what is the outcome? What is the result? Cell shape and size remain unchanged. The solution outside is isotonic. Cell shrinks in size and loses shape because the solution outside is hypertonic. Cell slightly enlarges or even bursts. The solution outside is hypertonic. When it is fully distended, that is firm, we call the cell as turgid. When it cannot withstand any further inflow of water molecules, it is going to burst. Now, in case of plant cell, another striking feature which determines the behavior when subjected to different is rigidity of the cell wall, which resists bulging and protects the delicate cellular parts inside. And this phenomenon related to this behavior are we have terms here, turgidity and plasmolysis and placidity, which you will learn later in this chapter. See again, this is the condition. What happens in the isotonic? No change, shrinks and swells again. In the plant cells, no change, shrinks. This shrinks, it is going to shrink the cell membrane, and this space between the cell wall and the cell membrane filled up with hypertonic solution. And here again, in hypertonic, water is moving inside and the vacuole become distended, and you can see that the cell membrane is now very close to the cell wall. So certain nutrients such as ions of nitrate, you can mark these nitrates, and then sulfates, potassium, zinc, manganese, they cannot pass through the cell membrane. This is because their concentration is higher inside the root cell, and it is so maintained in order to develop osmotic pressure for absorbing water. Okay, so they are more in the cell sap. That is why when we water the plant, the roots absorb water from them easily. The iron concentration inside the cell sap is more as compared to outside. So in this way, the concentration gradient of this iron is opposite to that of the diffusion. And to obtain these ions, the plant has to forcibly take them inside from their region of lower concentration to the region of their higher concentration, that is from outside from the soil to their higher concentration, that means toward the cell sap. And to do this, they require energy. And from where is this energy coming? It is supplied by the cell in the form of ATP. So any process which is taking ATP, that is known as active transport. Okay, so passive transport, where we don't require ATP, it refers to requiring no input of energy, free movement of molecules from their higher concentration to their lower concentration. So the difference between passive and active transport is active transport requires ATP and passive does not require ATP. And this progress check, you have to do it and just see whether you are clear till here. Thank you so much.